Every single day, your body pulls off an engineering marvel on a microscopic scale, literally trillions of times over. What is it? It's perfectly copying the entire instruction manual for life. So today, we're going to dive into the incredible molecular machine that duplicates your DNA. And when I say the entire manual, I am not kidding. We're talking about a genetic code that is over 3, get this, billion letters long. And every single time one of your cells divides, it has to make a perfect copy. No typos allowed. The stakes are incredibly high. All right, so here's our game plan. We're going to look at the sheer scale of this copying challenge, then see how the cell finds its starting blocks. We'll watch it assemble the molecular machine, kick it into high gear, and then cover the all-important one-and-done rule. And finally, we'll see what happens when this amazing process goes wrong. Okay, let's kick things off with the ultimate copying challenge, the blueprint for life. Seriously, think about the scale of this problem. This isn't just like copying a file on your computer. We're talking about flawlessly replicating the blueprint for you. This has to happen for you to grow, to heal a simple cut, even just to replace old cells. The need for precision is just immense. So that leads us to the big question, right? How in the world does a cell copy a library that's billions of letters long, do it super quickly and almost flawlessly, trillions of times a day? I mean, it sounds completely impossible. But as you're about to see, the cell has this, incredibly elegant solution. All right, section 2, finding the starting blocks. With any huge project, the first step is figuring out where to even begin. For DNA replication, the cell can't just start at one end and chug along to the other. That would take way too long. Instead, it's much smarter. It pinpoints thousands of specific locations called origins of replication. Think of these as the official starting blocks for this massive copying race. And this is the cell's secret weapon, really, massive parallel processing. To copy billions of letters in just a matter of hours, the whole process has to kick off at thousands of these spots all at the same time. It's kind of like having thousands of construction crews starting work on different floors of a skyscraper simultaneously. It's the only way to get the job done on time. But here's where it gets really interesting. How cells find these starting blocks isn't the same for everyone. Take something simple like yeast. For yeast, the starting point is like a very specific street address, a particular DNA sequence. Easy to find. But for us humans, it's way more complex. There's no single address. It's more like searching for a favorable neighborhood. The machinery looks for broader clues, like areas where the DNA is packaged more loosely, making it easier to unwind. Okay, so we found our starting blocks. What's next? Well, now we have to assemble the molecular machine itself. This is where something called the pre-replication complex comes in. Before any copying can even think about starting, this whole team of proteins has to gather at each origin. You can think of them as the prep crew, getting the site ready for the main event. So what does this pre-replication complex, or pre-RC, actually do? Well, its most important job is to license an origin. And you should really think of this like a one-time permit to build. Once an origin gets this license, it's officially good to go for replication. But, and this is absolutely crucial, it's only good for one round of copying per cell division. This is a major control point. So how does this machine get built? It's a very precise step-by-step assembly line. First, the surveyors arrive. This is a protein complex called ORC, and it lands right on a starting block. Next, it calls in the loaders, 2 proteins named CDC6 and CDT1. Their job? To bring in the heavy machinery. In this case, that's a ring-shaped enzyme called the MCM helicase. It's basically the unzipper. The loaders carefully place this ring right onto the DNA strand, and boom, the site is officially licensed and ready for action. All right, so the machinery is built, the sites are licensed, we've got thousands of these pre-RNAs just sitting there on the DNA, all poised and ready. They're just waiting for the green light. This next part is all about the go signal, flipping that switch from ready to replicate. And this timing is controlled with incredible precision by the cell cycle. All that assembly work we just talked about, that happens during a phase called G1. But then, once the cell enters the S phase, S for synthesis, a couple of key enzymes give the official go signal. This signal hits the MCM helicase, waking it up and telling it to start unzipping the DNA double helix. And once that DNA is unzipped, the real star of the show, DNA polymerase, can finally get in there and start building the new strands. Now, check this out, because this is just an amazing detail. The cell has this incredible foresight. It actually builds and licenses way more of these starting blocks than it usually uses. These extras are called dormant origins, and they're basically a backup plan. So what if the copying machinery hits a roadblock, maybe some DNA damage? No problem. The cell just activates one of these nearby dormant origins to pick up the slack and make sure the whole chromosome still gets copied on time. It's a brilliant built-in failsafe. So we've talked about getting the license to copy, but now we have to talk about maybe the most important rule in this entire process, the one and done rule. You have to make sure the genome is copied exactly once. No more, no less. If you accidentally copy even one part of your DNA twice, you're looking at total genetic chaos. It would be catastrophic. So the big question is, how does the cell enforce this incredibly strict rule? Well, it has a really clever two-part safety system. First, remember those enzymes that gave the go signal? Well, now they switch jobs. They become the project manager, and their new role is to prevent any new machines from being built. They do this by slapping a destroy me tag on key components like that CDC6 loader. And at the exact same time, another protein called geminin comes in. It's like the safety inspector. It literally just grabs onto the other loader, CDT1, and physically stops it from loading any more MCM unzippers onto the DNA. So you see, it's not just one safety measure, it's multiple layers of protection. You've got high CDK activity acting as a blanket no to new assembly. You've got key proteins being actively targeted for destruction. You have inhibitor proteins like geminin physically blocking the loaders. And just to be extra sure, once the MCM unzipper is done, it actually gets kicked out of the nucleus until the next cell cycle. The cell is absolutely not taking any chances here. Now, this whole system is incredibly elegant and robust, but it's not perfect. Things can go wrong. And this is where we connect all this microscopic machinery to something very real, human health. What happens when the parts of this machine are faulty? Because this process is just so fundamental, it's how you make new cells, even tiny errors in assembling this replication machine can have absolutely devastating consequences. It's pretty straightforward. If you can't build the machine right, you can't copy your DNA right, and if you can't do that, you can't make new cells correctly. That's going to impact growth and development right from the very start. And a really clear and frankly tragic example of this is a condition called Meyer-Gorlin syndrome. It's a rare genetic disorder, and we now know it's caused directly by mutations in the genes that code for these pre-RC components. The syndrome leads to some serious developmental issues, like dwarfism. And when you get down to it, the reason is simple, but profound. Their cells just can't multiply properly because the DNA copying machines are broken. And this is where it all comes together. The specific genes that are mutated in people with the syndrome, they're the exact ones we've been talking about this whole time. We see mutations in the genes for the ORC complex, our origin surveyors, and in the genes for CDC6 and CDT1, our machine loaders. It's a perfect illustration. When the parts that are supposed to find the starting blocks or load the machinery are broken, the entire project just grinds to a halt. So, when you think about it, from a 3 billion letter code to this incredibly complex machine with layers of failsafes, DNA replication is just a fundamentally awe-inspiring piece of biology. This elegant machine is copying the blueprint for life inside you right now. And it really makes you wonder, doesn't it? What other incredible secrets are hidden away in these tiny microscopic processes that literally make us who we are?